All right. Hi, everyone. Thank you all for coming tonight to our first workshop of the year on the basic biology of cancer. Awesome. Next slide. So before we start, we'd just like to acknowledge that today we're dialing in from the lands of the Wurundjeri people who have been custodians of this land for thousands of years. We'd also like to acknowledge and pay our respects to their elders, past, present, and emerging, and extend this respect to everyone who may be dialing in from different countries. So I invite you all to use the chat function to write a quick acknowledgement of country of your own if you'd like.
As you probably already know, UCCC is an initiative of Young Australian Cancer Initiative. We are a student-led, not-for-profit organization that aims to nurture a future generation of cancer-literate individuals who are empowered with the skills and knowledge to reduce the cancer burden in our society. The UCCC aims to provide an opportunity for university students across Australia to get together in groups of 2 to 4 to tackle a real issue in the cancer space. This year, the case challenge is to develop a public health action model for liver cancer awareness to increase early diagnosis and improve survival rates. On that note, it is my pleasure today to introduce our workshop facilitators, Qirui and Prageeth, who are also our research officers from the committee, as well as knowledgeable student doctors who are keen to teach you about the basics biology of cancer. So without further ado, I'll let you guys take it away.
Great, thanks so much for the introduction, Christy. So the objectives of this workshop is to just give a bit of a basic background behind cancer and liver cancer as well as hepatitis. So I'll be doing a bit about the basic biology of cancer, and Prageeth will talk a bit about a bit more specifically about hepatitis and liver cancer. So the level this is pitched at is quite basic. So it's meant for people who don't really have a science background or who haven't had much experience with the biology of cancer. So if you're a med student, a biomed student, or something sciencey, you've probably already heard this stuff. You might be more interested in Prageeth's section, which is a bit more specific. But yeah, my section is pretty basic. It's meant for more people who don't really have a background. So yeah, hi. So I'm Qirui. I'm a third-year medical student at Melbourne Uni. And before I start, I also want to acknowledge that I'm presenting from the lens of the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders, past, present, emerging, as well as any Aboriginal people who might be in the audience today. Yep. So these slides have been adapted from a high school biology masterclass that YACI also does. Great. Let's get started. Next slide, please. So firstly, what is cancer? So cancer is a group of diseases caused by abnormal cells that grow uncontrollably. So and the keywords here are abnormal cells, so these cells are not normal, as well as the fact that they grow uncontrollably, which means, cells grow normally all the time, but in a very controlled fashion. But what's significant about cancer is that these are abnormal cells and they grow uncontrollably. And to understand cancer, it's pertinent that we understand normal cell biology as well. Next slide, please. So this is just a bit of a background behind what cells are, if you don't really know yet. So all organisms are made of these little things called cells. There are many different types of cells that make up many different types of tissues. And sorry guys. And the different cells in our bodies also perform different functions. So you have cardiac cells, skin cells, muscle cells, different types of cells. Next slide, please. Great. So this is just some of the different cell types that we have in our body. We have epithelial cells, and a significant, an obvious example would be like skin. We have connective tissue cells, which makes up fat, cartilage, blood, and stuff like that. We have muscular cells, which we're all probably quite familiar with: skeletal muscle, smooth muscle that contracts on its own, and cardiac muscle, which is a special type of muscle cell, as well as nervous cells, which are our brain, spinal cord, nerves, and stuff. Next slide, please. Yeah, so this is a bit of a graphic that we've all probably seen in high school if we did bio. A cell has many components, but some significant components are stuff like the nucleus, the endoplasmic reticulum, lysosomes, Golgi apparatus, mitochondria, which we all know is the powerhouse of the cell. Next slide, please. So just to go a bit more specific into the different parts of the cell. The nucleus is the part of the cell that contains all the DNA, and DNA is the genetic material that makes all the proteins, makes all the stuff that we need for our body. An easy way to analogize this is that the nucleus is like the brain of the whole cell. Like it controls like all the cell processes and does a lot of important things. Next slide, please. So this is just a slide to show you what DNA is if you don't already know. DNA is this double helix structure thing that encodes for all the proteins in our body. Next slide, please. Yeah, and this is just a slide to highlight to show you all that DNA is made up of complementary base pairs. A pairs with T, G pairs with C, and vice versa. Next slide, please. So why is the DNA sequence important? So the DNA sequence is important because it is specifically ordered in such a way to build like the kind of proteins that we need. And if this sequence goes wrong, then wrong proteins are made and bad stuff happens. Next slide, please. So this is just a quick graphic on how DNA becomes proteins. So DNA gets unzipped, DNA gets copied, DNA gets read and becomes messenger RNA, which is transcription. I'm sure we're all familiar with mRNA. It's all in the news nowadays. The mRNA is then edited and it gets translated into proteins by the ribosomes. Next slide, please. So just as a quick summary of what I've gone through so far, there are different kinds of cells in different tissues, like the different examples that we went through. All the cells contain DNA, which is like the genetic component, the genetic library that makes all the proteins that we need. In terms of how DNA becomes proteins, DNA becomes RNA, and RNA then becomes proteins. And as you can see, it's quite a lengthy process, so that there's many areas that can go wrong. So, and when these things go wrong, then we get wrong proteins, and that's how like cell function becomes dysfunctional. And we encode for wrong proteins when the DNA sequence itself is wrong, when something goes wrong in the translation process of making DNA to proteins. That's how we get all these erroneous proteins. By the way, if I'm going too fast, or if you have any questions at all, you can just type in the chat. If you're too shy, you can just private message me or something. I'll try my best to answer them. Great. So next slide, please. So in terms of a normal cell life cycle, it exists in a few stages. So there's a G1 phase, there's an S phase, there's a G2 phase, which are not like of massive importance in terms of cancer. But what we really want to focus on is the M phase, the mitosis phase. Next slide, please. So mitosis is basically cell division. It's the process by which cells replicate themselves, and one cell becomes two cells basically. There's quite a few steps involved. So if you did high school bio, there's interphase, there's prophase, there's metaphase, anaphase, telophase, cytokinesis, I can never say this word, cytokinesis. Yeah, so as you can see, the whole point of this slide is just to show you that there are many steps involved and many things that happen for a cell to like replicate, for a cell to like divide. And when many things happen, the margin of error just gets bigger and bigger, and that's how things go wrong. Next slide, please. And also in terms of, things can also go wrong in terms of the cell signaling. So a cell isn't like a big functional living organism like us. It doesn't have a lot of brain power on its own. The only way a cell knows when to divide or how to divide is by getting signals from other cells. So as you can see, it's quite a lengthy process as well. They receive a growth factor or some sort of signaling. There's some sort of internal signaling called transduction, and then it results in that cellular response where they either make proteins or they divide or they do what they've been signaled to. Also quite a lengthy process. You can see like even transduction itself has so many steps. Reception itself has so many steps. There's receptor ligand binding, things have to like change and stuff. Yeah, okay, next slide, please. So that whole background is just for me to highlight the fact that this whole process has many steps, and because there's so many steps, many things can go wrong. So you can have the wrong DNA. So the DNA is erroneous to start with. You can have the wrong RNA, so translation goes wrong. You can transcribe the wrong protein, or even after you've done everything correctly, you can still get a very abnormal cell function if something else has gone wrong. Next slide, please. So we've talked a lot about how things go wrong, how there's many steps, wide margin of error. But what's actually what's actually preventing stuff from going wrong? So there are many mechanisms to help stuff not go wrong. So one of the mechanisms is that DNA is repaired by enzymes. So we do have enzymes that try to proofread the DNA, make sure stuff doesn't go wrong, make sure transcription, cell division, translation doesn't go wrong. Oh, that's not right. Yeah, but yeah, essentially. So there is a DNA repair mechanism within our bodies, and if too much damage happens to cells, they're meant to be able to kill themselves. So apoptosis, programmed cell death. So if too much damage accumulates, if too much stuff just goes wrong, there is a mechanism where the cell would just kill itself. So it prevents the cell from passing on all these errors. And we also have the immune system that acts as that provides some sort of surveillance to help us to destroy damaged cells, to promote apoptosis in problematic cells and things like that. Okay, next slide, please. Yeah, so in summary of the stuff I just presented, if you didn't take anything else away, you can just look at the summary slides. There are signals from inside and outside of the cell to start the cell cycle. There's reception, transduction, cellular response signals, and a lot of errors can occur in this signaling. So I guess the point I'm trying to bring across is that cell division, DNA transcription, and all this stuff like that are multifaceted, multi-step processes, and because they're so complex and so lengthy, there's a huge margin of error, and many things can go wrong. Even though we do have all those, we do have a lot of surveillance mechanisms, but it's just such a long process, and many long processes, there's just a lot of margin for things to go wrong. Next slide, please. Great. So in terms of basic, to come back to cancer a bit. So if you look at the first picture, it just shows normal cells, but sometimes like in all the stuff that I just mentioned, sometimes things go wrong, and then these cells become abnormal cells. The abnormal cells then multiply, and then they start invading into the tissue below. And these are all hallmarks of cancer. So things go wrong with our cells all the time. We have a lot of abnormal cells right now in our body, but the difference between us and people who have cancer is that our abnormal cells don't multiply uncontrollably, and our abnormal cells don't invade the tissue, the surrounding tissue, and don't become malignant. Next slide, please. So to better illustrate this concept, if you look at the histology slides, if you look at the top row, the slides are all really well ordered. I mean, just take for example the one all the way on the left, the epithelial cells. If you look at the one on the top row, they're really well ordered. You can see they are well structured. They don't look messy. If you look at the ones below, you can see they're all over the place. Just comparing the one above and below, the histology is vastly different. The cells below have gone all over the place. They're no longer as ordered. They're no longer as structured. And you can see that the basal layer is beginning to get invaded as the cells push into the surrounding tissue. Great. Next slide, please. So tumor is a really broad term, and you've probably heard of benign tumors and malignant tumors. And so what differentiates a benign tumor and a malignant tumor? A tumor just means a group of cells that are growing abnormally. And yeah, next slide, please. So this is just a graphic to show you the differences between a benign tumor and a malignant tumor. The in the top row, the picture on the left and the picture on the right are both tumors, but one clearly looks a lot worse than the other. If you look at the one on the left, it's like well circumscribed. It's contained. It's still a tumor, but it's not malignant or not malignant yet. If you look at the one on the right, it just looks significantly worse. Like it's not well circumscribed. It's all over the place. There's clearly a lot of invasion taking place. It's pushing into the surrounding tissue and everything. Next slide, please. So as I've mentioned previously, genetic damage in DNA leads to cells becoming cancerous, and that is what underlies cancer cells. It is something has gone wrong in all the processes I've previously mentioned. This results in accumulated DNA damage, and then it becomes the abnormal cells. Next slide, please. So this is a pretty key slide. So there are, like I said just now, there are many different kinds of abnormal cells. There are many different kinds of tumors. But what really makes cancer cancer? Like it's a benign tumor cancer. It's having abnormal cells cancer. So just to holding on this point, a couple of guys came up with this hallmarks of cancer thing. And if you just look around the wheel, there's sustained proliferative signaling, evading growth suppressors, blah blah blah. And I'll try to go through a few key ones. But I think they started with like 6 or 8, and then they added a few more as they realized that cancer cells were evading immune destruction and stuff like that. But what really underpins this whole thing is the fact that cancer belies genetic instability and the fact that they're all uncontrolled, and the fact that it's uncontrolled genetic instability. Right. Next slide, please. So just as a bit of revision, so like I've said just now, like I've shown just now, that there is a cancer, there is a cell cycle, and this is the graphic for like cell division. Next slide, please. And within the cell cycle, there are important checkpoints at G1, one at G2, and these checkpoints are there to make sure that the cell doesn't progress past the checkpoints if something has gone terribly wrong. So for example, if something has gone wrong before the G1 checkpoint, the G1 checkpoint is meant to be there to make sure that the cell doesn't progress beyond the G1 checkpoint and go to GM or G2 or S, the S phase. So essentially, I mean, that all sounds a bit complicated, but essentially these are just inbuilt cellular mechanisms that help to regulate and prevent mutations from accumulating. So we do have that kind of stuff, like I said just now. Next slide, please. But the problem with cancer is that cancer bypasses all these checkpoints. So even though we have a lot of mechanisms to stop cancer from getting out of control, cancer manages to evade all these mechanisms and just proliferate uncontrollably. And that is the issue with cancer. Next slide, please. Yeah, and one of the big issues with cancer is that they basically just take over the cell cycle and they just run their own cell cycle. And they do this by sustaining growth signals. So they produce a lot of growth signals that help themselves to grow. And they also evade all the stop signals that the body tries to, so the body tries to stop all these malignant cells, but they can't, because the cancer cells have evolved mechanisms to evade all our stop signals, such as our checkpoints, our regulatory enzymes, or whatever. Okay, next slide, please. So sustained growth signaling is a hallmark of cancer, and what it essentially is that these cancer cells are really good at making growth factors for themselves and growth signals for themselves, and they just keep growing and growing and growing. Yep, next slide, please. And they also manage to escape the growth suppression from the body. So like we were talking about just now, there are many mechanisms and there are many like regulatory checkpoints and stuff. But what cancer manages to do is to evade all these mechanisms. And one of the key ways they do this is by damaging the tumor suppressor genes. So we have genes in our body that are called tumor suppressor genes, and I mean, like the name says, one of their main roles is to suppress tumors from getting out of control. But cancer that becomes malignant manages to successfully evade all these tumor suppressors or damage the tumor suppressors. And because of that, the tumor suppressors don't work as well, and the cancer manages to escape all these growth suppression. Next slide, please. So another key hallmark of cancer is also invasion and metastasis. So as we were talking about in the benign versus malignant example just now, like what makes a tumor benign and what makes a tumor malignant, metastasis is a key point. It's actually a key hallmark of cancer. So if you have a tumor and it's not malignant, you can hardly call it cancer because metastasis is characteristic to cancer. It is when the tumor starts invading surrounding tissue. It starts metastasizing to other tissues, other organs. It starts invading surrounding tissue. That is when it becomes malignant. That is when it becomes cancer. Next slide, please. Right, and this is just another one of the hallmarks. So it's called replicative immortality. And what basically happens is that there's actually in most of our body cells, there is actually a limit to how many times they can divide. So the body has inbuilt mechanisms to stop these cells from dividing uncontrollably. And for those who are interested, at the end of DNA, there's a little thing called a telomere. And every time the cell divides, this enzyme called telomerase cuts off a bit of the telomere. And once the whole telomere is gone, it is a signal for the cell to undergo apoptosis and just destroy itself, so that it doesn't keep replicating. But what cancer is able to do is that it can prevent this cutting of the telomeres, and because of that, they can keep replicating, and a key signal to stop this uncontrolled replication is lost when the cancer disrupts this cutting of the telomeres. Next slide, please. Yep. So this slide looks a bit messy, and those are pretty big words. But what angiogenesis basically means is to is to form new vessels, new blood vessels. And what energetics basically means is that cancer cells thrive on taking up glucose. They thrive on aerobic they thrive on a certain type of aerobic respiration that helps them to take up super lot of glucose. And to put it simply, angiogenesis just means cancer cells create their own like vasculature. They create their own like blood supply, and because of that, they get a lot of nutrients, and the nutrients help them to grow even faster, help them to divide even faster. Yep. And cancer cells also manage to avoid apoptosis. So like we talked about just now, apoptosis is it's programmed cell death. Is where cells can kill themselves if too much metabolic damage is accumulated. But what cancer cells manage to do is that they manage to avoid this whole step. And if you see the P53 P53 in the slides, P53 is actually a tumor suppressor gene like we talked about just now. So in normal cells, if something goes wrong, P53 should be able to kill it and hence prevent tumors from forming. But what cancers can do is avoid P53 and hence help them keep replicating accumulated damage. And it just creates more uncontrolled cell replication. Next slide, please. So as we talked about just now as well, the immune system also provides surveillance for cells that are damaged and stuff like that. But cancer cells also manage to avoid immune detection and immune destruction. So I think a couple of years ago, the Nobel Prize was actually for immunotherapy against cancer cells. So that is a field they're trying to work on. But currently, a lot of the cancers just escape immune detection, and the immune cells can't kill these cancer cells. So that is a massive problem as well. Next slide, please. And finally, one of the key hallmarks is also genomic instability. Like we talked about just now, if the genome is stable, if the DNA is stable, if everything is stable, we shouldn't be replicating replicating and dividing cells that are abnormal. But one of the key hallmarks of cancer is that these cells do have genomic instability, and hence it replicates into a lot of problematic cells. All right, next slide, please. Right, so just as a bit of a summary, so there was quite a whirlwind for all the hallmarks, but just as a bit of a summary, not all tumors are cancerous. So we discussed we discussed the difference between benign tumors and malignant tumors. What makes a tumor benign? What makes a tumor malignant? And it is the malignancy and the metastasis that is key to cancer formation. And not all damages to DNA also will become cancerous. So like we talked about, there's a lot of immune checkpoints, there's a lot of cell cycle checkpoints, there's a lot of regulatory mechanisms. But what cancer manages to do is avoid all these mechanisms and keep replicating their problematic cells. Brilliant. And so next slide, please. So for those of us who don't know much about immunology, this is just a bit of a crash course of immunology. I wouldn't say it's pertinent to understanding cancer, but just because a big part of cancer is avoiding immune destruction, I thought it'd be good to touch on a bit of basic immunology. So next slide, please. So a role of the immune system is to be able to recognize cancer cells that we talked about just now. And because they're abnormal, the immune system tries to attack cancer cells. Next slide, please. So the immune system is basically made up of an innate immune system, adaptive immune system. So the innate immune system is stuff like physical barriers, like our skin. So our skin forms this layer around our body to prevent germs from getting in. We have chemical barriers, like we have stomach acid that destroys bacteria in the food that we eat. We have mechanical barriers as well, like tears and stuff like that. In the second line of defense, we also have like little cells that if the pathogens do manage to get into our bodies, we have little cells that destroy these pathogens. We also have defensive molecules, and we have an adaptive immune system, which are basically T cells and B cells. And that's what's happening when you get a COVID vaccine. The COVID vaccine activates the adaptive immune system to form memory, so that you can fight the COVID pathogen. Next slide, please. Yeah, so this is just a brief overview of the cells and the immune system. So cells like macrophages, neutrophils, make up those second line defense cells that kill any pathogens that get into our body. And we also have B cells, T cells, which are the majority of the adaptive immune system, as you can see. Next slide, please. So the key role of B cells is to make plasma cells, which makes antibodies. Essentially, like I'm sure you've heard of the term antibodies. So these antibodies come from B cells that become plasma cells that produce antibodies. And the role of the T cells to make helper T cells, as the name suggests, helps the immune system. So it provides a complementary output to what the immune system is already doing. And cytotoxic T cells, and if you can't guess from the name, cytotoxic T cells basically do direct killing of the pathogens, the bad guys in our body. Next slide, please. Yep, so if you can't remember anything else about B cells, just remember that B cells make antibodies. And next slide, please. And T cells kill the pathogens, they kill the bad cells. Next slide, please. Great, so just to summarize, like the stuff that we've gone through. The nucleus stores protein encoding genes in DNA, DNA becomes RNA, and which becomes proteins, and there's a massive cell cycle with many steps. There's a lot of cell signaling involved as well, and because there's so many steps involved, there's a wide margin of error that sometimes leads to errors in DNA, which leads to dysfunctional proteins. And it is these dysfunctional proteins and these problems in the cell cycle that can lead to cancers. Essentially, cancer is a genetic disease of abnormal cells that grow and divide uncontrollably. So like we've gone through, we do have many abnormal cells, but it is the mechanisms in our body that prevent these abnormal cells from growing and dividing uncontrollably, that prevents us from getting cancer. And when these mechanisms fail, like all the things that we went through just now, then these abnormal cells start growing and dividing uncontrollably, and that's when we get cancer. And just as a final note, our immune system normally recognizes these abnormal cells and tries to attack them, but a key hallmark of cancer is that cancer is able to evade the immune system and evade the processes that try to destroy it. So that's what makes it so problematic. Great, thank you so much for listening to me. I'll pass the time on to Prageeth now.
All right, thanks so much, Qirui. Next, I'll be discussing hepatocellular cancer specifically as one of the types of cancers and the cancer that's related to this case, as well as hepatitis and the hepatitis virus. I'll just share. Make sure, can that be seen?
Yep.
Yeah, perfect. And if at any stage anyone has questions, we'll probably get to them at the end of the presentation, but feel free to add them into the chat or even direct message me or Qirui, and we'll try and address them at the end. So we'll be discussing hepatitis in this presentation. First, we'll start off with liver anatomy or basic liver anatomy. We'll discuss what hepatitis is and the complications of hepatitis and the complications of liver infections. Furthermore, we'll discuss what hepatocellular cancer is. Then also, what is the significance in the Australian context and in the healthcare system? And finally, we'll end the presentation. So liver anatomy. First, we'll discuss what the function of the liver is. So the liver is an organ that's located in the upper right quadrant of the abdomen. You'll find it right underneath your ribs on the right-hand side, on your right-hand side. So the liver has multiple functions. So the main function is it can synthesize bile, but it also, one of the functions it can synthesize bile, but it also is involved in storing glycogen, which is used for our energy storage. It produces enzymes that allow it to remove poisonous substances and drugs and toxins from our body. And it's also involved in the production of blood components and other things that are important to maintain our blood and our immune system. When we look at the overall structure of the liver has 4 lobes, and it has 2 main blood vessels that supply with blood or take the blood away. So we have the hepatic artery, that divides in the right and left hepatic artery, the hepatic veins, which take blood away and send it back to the heart, as well as bile ducts. And that's used, the bile is another component that we make in the liver, and it's used to help our digestion. However, the microscopic structure is probably more important when we consider what are the impacts of cancer or the impacts of hepatitis. So the microscopic structure of liver is very specific and very sensitive to changes. It's composed of cells known as hepatocytes. Those are the liver cells that make up most of the liver, as well as small arteries known as arterioles, small veins, and a small branch of the bile duct. When we consider, we'll come back to this microscopic structure later and to see how it can be impacted with different diseases. So next, what's hepatitis? Hepatitis in general is the inflammation of the liver tissue. Inflammation is an immune reaction that occurs in any of our organs or in our tissues, in this case, in the liver. The most common cause of hepatitis is the hepatitis virus. And there's 5 types of hepatitis viruses, A, B, C, D, and E. There are also other things that can cause hepatitis, a virus known as CMV or cytomegalovirus, Epstein-Barr virus, but otherwise alcohol use and fatty liver is also certain things that can cause hepatitis that you might know about. But specifically, we will discuss today what hepatitis is, mainly because of its relevance to hepatocellular cancer. And specifically, we'll be talking about 2 types of hepatitis, hepatitis B and hepatitis C. And we'll be discussing these 2. It's because these are the ones that cause chronic disease and have a large burden in causing hepatocellular cancer. So there are 2 viruses we're discussing, hepatitis B and hepatitis C. It's important to compare the difference and understand why hepatitis B and hepatitis C have different kind of effects on the population of the people that are infected. When we look at the percentage of people who are infected, about 0.9% of the population in Australia are infected with hepatitis B. So that's about 9 in every 1,000 people. While hepatitis C, there's around 4 in every 1,000 people have hepatitis C in Australia. Hepatitis B is a DNA virus, while hepatitis C is an RNA virus. The transmission, or how you get hepatitis B, is mainly from blood and body fluids. Unprotected sexual intercourse is the most common cause of getting hepatitis B. in both developed and underdeveloped nations. But other ways of being transmitted include needle transmission. So this can include IV drug use and unsterile needles from tattoos or piercings. On the other hand, hepatitis C is not usually contracted from sexual transmission, and mainly it's again hepatitis C, on the other hand, is mainly gained from IV drug use or unsafe organ transplantations for patients. When we consider trying to manage hepatitis B, currently there's no cure for hepatitis B. On the other hand, hepatitis C has a range of antivirals that can actually cure and get rid of the virus in general. However, in untreated cases, only around 10% of patients who get hepatitis B have lasting chronic infections, which we'll discuss later. On the other hand, in untreated cases of hepatitis C, around 85% of patients can get chronic infections. And another key point that when we compare hepatitis B and hepatitis C is that there is a vaccine available for hepatitis B to prevent hepatitis B infections in the first place. However, there's no such vaccination for hepatitis C. So what are the kind of symptoms for hepatitis? We usually say that there's a triad of symptoms, or three main symptoms when someone has hepatitis, which is right upper quadrant pain. So that's pain where the liver is, fever, and jaundice. And jaundice is yellowing of the skin. Chronic hepatitis infections in general, otherwise, are the next step in hepatitis infections. There's acute and chronic. Acute is anything lasting less than 6 months, and some patients will just get hepatitis B for a few months, take treatment, and will no longer have hepatitis B in the body. However, some infections can evade the immune system and last more than 6 months. The symptoms usually die down in this period and make it hard to detect because it looks like that the initial hepatitis infection has resolved. However, over time, they may develop further signs, often of liver failure due to the ongoing infection. Other signs can include things like bleeding and bruising disorders, fatigue, poor appetite, jaundice, which is again yellowing of the skin, itchy skin, ascites, which is fluid buildup, especially in the abdomen or in the lower legs, as well as weight loss. It can also include many non-specific presentations, just fatigue for no reason, tiredness, excessive sleepiness, low blood pressure, etc. Next, we would like to discuss cirrhosis and hepatocellular cancer. So what's cirrhosis? And it's often related to with hepatitis and also with alcohol use. What is cirrhosis? So cirrhosis is the permanent and irreversible diffuse scarring of the liver. Now, scarring and fibrosis of the liver is usually normal. So if there's fibrosis is what occurs when is a method of repair by the body whenever there's damage that occurs. When we cut our skin and you see areas of skin that look different to around the other parts of the skin, that's because that's again a form of fibrosis. However, the problem in the liver is that after a while, if fibrosis is continually occurring, it can have impacts on the architecture of the liver and prevent the normal liver function. If you can think about it this way, if there are normal liver cells that die, and the way that the body replaces it is with connective tissue, i.e. that's tissue that maintains the shape of the liver, but it doesn't provide the same function to the liver. So it doesn't do the bile production. It doesn't produce the blood contents. It doesn't produce the enzymes that the liver will normally create. This can result in a disease because it impacts the long-term liver function. So how does hepatitis then cause hepatocellular cancer? This is another aspect of how hepatitis replicates. So hepatitis infects cells by incorporating its DNA into the normal liver DNA. But a consequence of this is that if the DNA is damaged or continually impacted by infection, as Qirui mentioned in the previous things, this can impact the stability of the DNA and the normal functions of the DNA. And it might result in the DNA gaining those hallmarks of cancer and resulting in the development of liver cancer. And so these cells, if they're able to evade the immune system and they're not able to resolve quickly, this can result in continual reproduction of those cells and the development of a tumor or a cancer. So this diagram again highlights how hepatitis could result in the development of a cancer. So when we look at the second step, when hepatitis replicates, it incorporates its DNA into the affected cells. After a while, and after continuous genetic accumulations, a cell might be precancerous. If it continues to develop more kind of mutations, these cells might replicate, and because these cells do not do the normal role of the liver, it can impact the liver's function. Next, we'll discuss what liver cancer is. So hepatocellular cancer is, again, any cancer that occurs within the liver and often originates from hepatocytes, the specific cells that are in the liver. And the development of cancer can impact the functions of the liver and have broad effects on the whole body. So what are the main causes of hepatocellular cancer? Well, around 50% of hepatocellular cancer is caused by a chronic hepatitis B infection. Another 35% is caused by chronic hepatitis C infections. So in total, around about 85% of hepatocellular cancers are due to either hepatitis B or hepatitis C infection. The other causes, it can include things like alcoholic cirrhosis and other things like family disorders like hemochromatosis, glycogen storage disease, and other causes like fatty liver syndrome. So the rates of liver cancer in Australia is around 7 per 10,000. That means that around about every year, around 3,000 to 4,000 people have liver cancer. So what are the symptoms of liver cancer? Well, liver cancer has, the problem with liver cancer is that it can be asymptomatic or have no presentation for a long duration. Because the mass is just growing and hasn't, if it's not impacting the liver, it doesn't mean there's no cancer there. It just means that it hasn't grown to the extent that it causes any effects on the liver. The other things that liver cancer can cause are things like liver failure signs. These are signs or symptoms that occur when the liver isn't working like it normally does. And that includes, again, things like pain in the liver area, yellowing of the skin, ascites or fluid accumulation, and skin changes. Often this is the things like veins being visible from the skin that shouldn't normally be visible. Otherwise, a key symptom of cancer is weight loss. If you can imagine a new mass of cells within your body, you can understand that those cells will take a lot more energy than all the other cells around it. Well, as Qirui mentioned in the last presentation, cancer cells often develop more blood vessels and often have a fast rate of growth. And because of that, it often takes the nutrients that will go to the rest of the body to those cancer cells. And that's why many patients with cancer will present with unexplained weight loss, i.e. they're losing weight because their body's using more weight than usual, even though they're not on a diet or they're not trying to deliberately lose that weight. So these are just two pictures of kind of the symptoms that can occur with hepatocellular cancer. On the left, we have what's ascites. This is a fluid buildup in the abdomen area. This is due to two main reasons. One is the liver produces proteins that keeps fluid in the blood vessels and stops it from leaving the blood vessels. But if the liver is not producing that chemical, which is often a colloid protein, the fluid can leave blood vessels and it can accumulate in the skin or in the lower legs. The other thing is thing called jaundice, which is yellowing of the skin, which you can see on the right. One of the functions of the liver is to get rid of toxins. In normal amounts, there's a chemical in the body called bilirubin that is filtered by the liver and it's removed from the body. However, when the liver is not working, that bilirubin can build up in the body and it deposits in our skin, as well as our eyes and sometimes other areas of the body as well. And so these are the symptoms that we see, which can suggest to us that a patient has either liver failure or liver cancer. So how do we go about diagnosing liver cancer? Well, the first thing we'd mainly do is something called antibody test. This test is looking for a reaction by our body against hepatitis viruses. If the test is positive, i.e. if we develop antibodies to a hepatitis, it suggests to us that there's still hepatitis within the body. Other tests that we can do is something called an alpha fetoprotein. This protein is usually in a low amount within our body, but if there is a cancer, it often goes, often the levels of alpha fetoprotein are elevated, and this may be the first sign or first kind of test positive that shows that there's a liver cancer specifically. Otherwise, there are tests called liver function tests. that look at the normal production of proteins in our liver. And where there's disease, these liver function tests can show abnormal results. Again, highlighting or telling doctors or whoever's looking at the investigations that there might be something wrong going in the liver. Other tests we can do are blood tests, because the liver has produces different things that are involved in the blood. And so if the blood tests are abnormal, it suggests that the liver could be one of the reasons why the blood's abnormal, as well as something we call an ultrasound. Ultrasound, you've probably seen, it's just that imaging thing. We often use it in babies to find out, in pregnant women to see if they're pregnant. But it can also be done around the abdomen, and it looks at if there's a density in the liver. Otherwise, MRI and CT produce images that can help us show if there's a cancer in the liver as well. And the management for cancer is often first line is surgery. Often we'll just try and go into the liver and try and take that tumor out, especially if it's non-malignant or benign, as Qirui has explained. However, if that cancer has then been able to leave the liver and gone to other areas of the body, commonly the lungs or even the bone and the back, then we will consider things like chemotherapy, which involves it's a drug that we administer the cell. Otherwise, in West, the cancer can't be cured because it's gone too far around the body, and so we're just managing the kind of symptoms of the liver cancer. So the prognosis for liver cancer is actually not as great as some other cancers that are out there, with the 5-year survival rate for even early stage hepatocellular cancer or liver cancer around 70%. And if it's a late stage or if it's a developed liver cancer, only 20% have a 5-year survival rate, i.e. that's the amount of people after 5 years who you'd expect to have not succumb to at least in 2021, there were 2,187 people who died from hepatocellular cancer. So next, so what's the significance of hepatocellular cancer? And this is kind of thing that you'll be trying to create an intervention for in the case file. Cancer has a significant care interventions. Approximately 226,000 and 189,000 people are estimated in Australia to have either hepatitis B or hepatitis C. However, many of these people are unaware that they're infected, and so they don't go to hospitals, they don't get the diagnosis, they don't take the are not taking medication, and therefore are at high risk of developing hepatocellular cancer in the future and other diseases, other kind of symptoms related to the infection and disease. And in general, it's also known that it can be a burden on the economic and kind of healthcare costs due to hepatocellular cancer, with around $139 million spent trying to treat patients with hepatitis and hepatocellular cancer. As we see from this graph, we can see this is highlighting the point we made in the previous slide, which is that unlike other cancers, the incidence or the amount of people who have hepatocellular cancer and the deaths from liver cancer have increased and don't show any signs of kind of lowering. So what are the main interventions or preventions we have? Well, for hepatitis B, which is one of the biggest causes of hepatocellular cancer, there is a vaccination that exists. Often it's 3 vaccinations, and this is administered to people. It's in the childhood immunization schedule. And because of that, Australia's been relatively successful in immunizing most younger children, with currently almost every 2-year-old, around 96% of all 2-year-olds being immunized for hepatitis B. On the other hand, for hepatitis C, there's no vaccine. However, there is antiviral treatment, which can cure around 95 to 97% of hepatitis B cases. However, the problem is that many people who have hepatitis C either are not following up on the treatment or not getting diagnosed with hepatitis enough. And so therefore treatment uptake of the people who have hepatitis C is only around 39.5% or 40% of individuals. There are also many barriers that prevented this kind of uptake of treatment or the uptake of immunizations. The first one is stigmatization. As you probably can understand from how hepatitis is gained to how people are infected with hepatitis, it's often from highly stigmatized routes, IV drug use, or from sexual transmission. And so individuals in these kind of groups or who have gained it from this are often very reluctant or can be reluctant in seeking investigations from doctors or discussing it with doctors and seeking treatment for these conditions. Otherwise, there are challenges in kind of healthcare and the management of liver cancer in different areas, especially regional remote areas, with around about 72% of people more likely to die from liver cancer if they live in regional and remote areas compared to Australian cities. Additionally, ethnicity and language remains a high cause. Even though 96% of people in Australia, 90% of Australian children less than 2 are immunized for hepatitis B, there's a large proportion of individuals outside of the country who might end up migrating to Australia that have not been immunized for hepatitis B or are at risk of hepatitis B. And so the main group of people who are actually infected with hepatitis B in Australia are those who have come from overseas. Additionally, we find that there is a poor health follow-up with people who have hepatocellular cancer or with hepatitis as well, with only around 27% of people adhering well to the optimal treatment, while most other individuals this graph again highlights what we've maintained in the previous slide, which is that most people who have hepatitis B, chronic hepatitis B in Australia, are actually those who were born outside of Australia, from Northeast Asia, 23.1%, Southeast Asia, around 20%, and people in Europe, 8%. And very few, even though there is significant portions who have gained it from IV drug use or from sexual transmission or other transmissions as well. So in conclusion, hepatocellular cancer remains a very significant cause of mortality and morbidity in Australia. And it's still, and hepatitis infections remain a significant cause of hepatocellular cancer. And in trying to have an impact on this hepatocellular cancers, the rates, mortality and morbidity that exist in Australia, there are a broad range of challenges and barriers that exist that have to be addressed. So finally, I guess that comes to the end of the presentation on it. We'd like to wish you all good luck. If there's any questions, please let us know in the chat. Otherwise, there's a few key dates that we would love to go through as well.
Awesome. Thanks so much, Prageeth and Qirui, for sharing all your knowledge with us and teaching us all about liver cancer and the biology of cancer. I hope everyone here learnt a bit from that. And yeah, if you have any questions, please feel free to text them to me or anyone else and we can answer them at the moment. And if not, you can also contact us in email and we can answer your questions after as well. Yeah. I don't think we have any questions at the moment. Maybe Christy, if you want to close up for us.
Yeah, sure. So thank you so much for Qirui and Prageeth and all our participants today for coming along. This has been a really fantastic workshop, and I'm sure everyone has gotten a lot of it. A gentle reminder to all the participants that the registration is Sunday on the 21st of August, and the submissions are due on the Monday, the 29th of August. Thanks, everyone.